Assalamu alaikum. Hello everyone. My name is Dr. Farhan Haq and I'll be teaching you Introduction to Bioinformatics. We will go through different topics of bioinformatics, concepts, where bioinformatics is used, why bioinformatics is important for biologists, and so on. There will be a lot of questions and hands-on experience trainings which we will go through during the process. So let's start from the history of bioinformatics and the definition. You all have heard about read the definition about that information processing, manipulating, and analyzing the data. That is more or less the comprehensive definition. But how that definition was introduced and how we came up with that definition, let's start from the first slide here. You can see that there is a name of a Dutch scientist, Pauline Hoogeweg, and she actually claims that she has introduced the term bioinformatics because when we go through if you go through the different articles published by Pauline Hoogeweg, you will understand that she actually was a theoretical biologist and she was trying to analyze the biotic systems. And from biotic systems, you know what is a biotic system? That the living things, all the living different things in an ecosystem that may include plants, animals, microorganisms. And when she was analyzing different systems and she wanted to compare those systems, so she came up with an idea that there should be some process to process this whole information because the data from microorganisms, from plants and animals, so there should be a comprehensive way where we can process all this information. So she came up with an idea that there is a biology and then there is an information technology. So biology and information technology, but she did not use the exact term bioinformatics, but her articles clearly indicates that she has an idea about how much the information system is important in biology. So recently she published an article in PLOS and she explained that actually I am the one who introduced this terminology, right? So that depends. There are a lot of different scientists as well who were working at the same time, but her articles actually indicates that she had an idea about bioinformatics. So now if we go to the brief history of bioinformatics, what actually is the importance here that you can see that how the data was accumulating and how the history converted or evolved into a time where the need of data science and bioinformatics become inevitable. So here you can see that if we start from 1953, and in 1953, you all know that Watson and Crick proposed a double helix model of DNA based on X-ray data. And from that day, actually, why this uh, discovery was important, uh, because from that 1953 and so on, if you go through the history after 1953, the data of different DNA sequences were introduced, then different protein sequences, amino acid sequences, RNA sequences were generated or discovered, and that whole process leads to a lot of different data sets from different directions. So they needed a comprehensive way to analyze that data. So if we go through one by one, in 1953, Watson proposed the double DNA helix model. And then in 1955, after just 2 years, the first protein sequence was analyzed and sequenced, that was bovine insulin. And that was done by Frederick Sanger. And he's a very famous scientist. And there is an institute with the name Sanger Institute in UK, right? So then after from 1955 to 1969, there's the whole 13, 14 years, and there are a lot of different DNA sequences were generated or discovered, and then a lot of different protein amino acid sequences were analyzed and sequenced. So then the scientists had a challenge of analyzing that information together. So some information was available, let's say, in Sanger Institute in UK, some was present in Stanford University in USA, some was in the University of California, some in some Asian countries, some European countries. So they needed a way to analyze all that information and have some common ground where they can analyze that. So one of the basic idea which came up between the two USA organizations, that was Stanford University and UCLA, they came up with an ARPA network they created to link the computers between between Stanford and UCLA, so they can share the information and they compare the information of that DNA and protein sequences. And here you can see that now the computers and the network became more and more important. And after that, you can see that in 1970, the first algorithm for comparison of two different sequences was introduced by two scientists, Needleman and Wunsch. They came up with an idea that how we can compare the two sequences through an algorithm. Obviously, this when the sequences were evolving or generating or this, then at that particular moment, they came up with an idea that we cannot analyze each sequence one by one or manually. There should be some algorithm, and Needleman wants to come up with an algorithm. We will study this algorithm later on. And then in 1972, the first DNA recombinant molecule was generated by Paul Berg and his group. So you can see that just in the span of 20 years, there was a lot of different information that was generated at protein DNA level, and there were a lot of different algorithms were also, coming our way. And then in 1973, you can see in just 20 years, there was a need of one database that is now still to date it is used as one of the most important database of protein, that is named the Brookhaven Protein Data Bank. And then you can see that now this database was contained all that sequences which were we started generating from 1955 till 1973, and till date a lot of different protein structures which are predicted are stored in this protein data bank. Then after that in 1988, uh, this was quite logical that when we have a lot of different sequences, protein sequences, then the scientists came up with an idea that why we sequence one gene or one sequence, one protein, why not we sequence the whole human genome and come and start analyzing the whole genome altogether. And for that, in USA, National Center for Biotechnology Information, in National Cancer Institute, they established started a new human genome initiative, and they proposed that in hopefully in next 15-20 years they will sequence the whole genome. And then in 1990, you can see that BLAST tool, which is till today it's the most widely used basic local alignment search tool. Why this became important? Because there are a lot of different databases and there are a lot of different sequences which were already available in different websites and databases, and they wanted to compare those sequences together using some method of alignment and come up with new ideas that how these sequences are related. And then in 1994, between 1990 after 1990, there were 1, 2, 3, 94, 95, 96. There were a lot of different databases. One example is of protein motif database, that is Print's database that was published by Atwood and Beck. There are a lot of more motifs. I just wrote one to show you that how from one DNA sequence to protein sequence to networks to databases, we have now analytical tools of analyzing different sequences and then comparing those sequences and coming up with new motifs, protein motifs and predicting motifs. So then in 1996, just in the span of after 1998, the Human Genome Initiative, 1996, scientists, before even publishing the whole human genome, they published the genome of Saccharomyces cerevisiae, that is Baker's yeast, and that was sequenced and published. It's very interesting to see that actually the yeast genome sequencing started after the Human Genome Initiative, but it was completed before the human genome because the genome size is very small, and that is only 12.1 MB. And as you know that in 2001 or 2000, roughly the human genome was published and its genome size was more than 3 GB. And they completed this genome before time, estimated time, because that was according to their first article that was expected to be finished after 2005, but they completed it in the beginning of the 21st century. So the 21st century started with the Human Genome Initiative. Now, this is the definition which I was talking to you about, the journal Bioinformatics definition. That is, bioinformatics is about searching, managing, and analyzing large amount of biological data using different computational approaches. So, So you can see that this bioinformatics actually deals with the biological data, and there are a lot of different fields in biology which needs bioinformatics for whole analysis. For instance, here you can see that there is one uh, this one term, forensic genetics, right? Then there is database. I have talked about a small database of protein data bank, a small example, and we will go into the detail about different databases in bioinformatics, their their importance and how they are managed. Then there's another field in bioinformatics that is named drug designing. For drug designing, for instance, there are a lot of protein sequences which are sequenced, and there are protein structures which are actually predicted and validated using different NMR, nuclear magnetic resonance, or X-ray crystallography, mass spectrometry, and then using different bioinformatics approaches. If we have one protein structure, known protein structure, and known drug, we can build a artificial environment and simulate the interaction between the drug and the protein and come up with a very good idea that is close to the nature. So what helps, and then the next is genome annotation. You can annotate the whole genome using bioinformatics approaches, starting from first gene to the last gene, or the first chromosome to the last chromosome. What are the functions? What are the characteristics? What is the significance? Anything you want to do about the genome annotation analysis at expression level, at DNA level, or at a protein level, whatever. Then modeling and interaction. These all different fields. I will talk about them in later in the lecture in the later lecture series. Here you just should have a brief idea about different fields in bioinformatics and how why they are important in managing those information. Then this next slide to the right of this uh, figure, you can see that there are different studies like proteomics, metabolomics, the next generation sequencing, microarray, epigenomics. All these are different terms, and we will go in detail. Like, for example, if we're talking about proteomics, it deals with the proteome data of a genome. Then metabolomics, we deal with the metabolomes. And then again, if we are dealing with the epigenomics, that is epigenome, that, for example, methylation and acetylation of the genome. So these things we will talk about later in the field. Here we are just talking about the different fields in bioinformatics. Then next generation sequencing, this is also another topic we will discuss in detail later on. Uh, uh, yeah. So now if you see here, these are different types of biological analysis, different methods. You have known, you may have an idea about in vitro biological analysis, in vivo biological analysis. In vitro, we within a controlled environment, outside a living cell, we analyze, for example, on petri dishes or in any cell line. We want to perform any analysis of any gene, or any check any mutation, or check the any expression of any protein. So we do those analysis using in vitro biology. And in vivo, within a living cell, we use different living organisms. The most standard animal is mouse models, and then there are also other models like zebrafish, so on. And now we always think that bioinformatics is something different, and we don't perform analysis or experiments, but you can call those also experiments. That in silico biology or in bioinformatics, we perform different experiments used on computers. So these you can say these as experiments. For example, if you want to analyze any expression of any gene, we download different genetic data sets and we perform analysis. If we want to see a structure, see how the protein structure folds, we use any protein methods to predict the structure. If we want to see the interaction between a protein and a drug, we perform different tools and perform our analysis. So these are different experiments. So we can also say these as experiments. And so there are three main types. So here All these are interrelated. For example, if we want to generate a we want to generate a hypothesis, what is very important in bioinformatics is that we should have any background knowledge or any hypothesis about any question of interest. For example, if we want to see that is there any association of ABC gene on any disease? So for that, first we in bioinformatics, first we perform bioinformatics analysis using any data set. There are a lot of databases which we will discuss later. We download that data set and then perform that analysis. We try to find that if the expression of that gene is varying in a disease. After doing that in silico, then we go to in vitro analysis and perform that on some cell line, and then we can also validate those on in vivo and controlled in any living cell. So these all different methodologies in bioinformatics, biological analysis are interrelated. So we should have, even if we are a biologist or we are a bioinformatician, we should have an idea about all these methods and how these are interrelated. So types of experiments in bioinformatics. Bioinformatics isn't just about storing the biological data in databases. It also concerns conducting experiments on that data, right? So if we are uploading any data on the databases, it means that data has any biological importance. So for that, we perform different experiments on that, and we try to come up with different uh, ideas that how this data is working on in any biological in any environment. So there are multiple types of methods, like searching is also an experiment. We want to search, comparing different genes, modeling the gene, and integrating that information from different sources. So we say that this is actually we say that these are different experiments. For example, what does searching mean? Let's suppose the simplest of all, this picture is taken from EBI website. Here, if you want to search, for instance, we want to search a protein sequence, then we have to go into protein databases, and then we write that gene and then search that gene to find any important information about those protein sequences. If we want to search for the nucleotide sequences, we go to DNA databases and try to find that sequences. And again, any enzymes we want to look at, whether there's any enzymes, whatever we want to search, we go into enzyme databases. There are databases, small molecules like different drug targets we want to find, then we go to small molecule database. Then if you want to review different our questions, we go to literature. So just like we search samples, just like we generate primers, sequences to make to perform analysis, in bioinformatics we also perform this, the simplest of all experiments, that how we can search uh, any biological molecules, biomarkers, using different databases. So this is the simplest of all experiment. Then if you want to compare, this is also one sort of an experiment. For example, we want to compare the sequence of multiple genes for a single gene in multiple species. Let's suppose evolutionary analysis. We want to see that how much the sequence is conserved. For that, we have to perform any comparative analysis between different species, that whether that sequence is conserved between different species or there is any variability. Just like if you see here, we have performed an experiment here that we are comparing histone H protein between human, mouse, rat, cow, and chimp. And we, after comparing between different species, we identified that there are some sites which are conservative between all throughout the species, but there are some which are not that conservative and they are non-conservative. It means that there is a variability in this site. And similarly, if we are comparing any, we want to analyze any disease, that whether why the diseased patient is showing different expression of any gene, or whether there is any specific mutation due to any disease. We compare the sequence of that gene and find any site which is possibly mutated between different species. So this is also a sort of an experiment, and in experiment we are comparing, just like we try to do polymerase chain reaction, and then we perform any mutation analysis and try to find whether that gene is mutated or not. in bioinformatics analysis we do these type of experiments. So in the next part we will go through the other types of experiments later on. Here I think for now on this lecture is concluded here, and let's meet with the new topic with the next type of experiment, that is uh, integration and all. Thank you. Allah Hafiz.